Hey everyone, we will now be discussing the second part of epistasis. So these are the different types of epistasis. So to start, let's just differentiate dominance and epistasis. When you say dominance, this involves intra-allelic gene interaction and that one allele hides or masks the effect of other allele at the same gene pair. If it's epistasis, it involves inter-allelic gene interaction and that one gene hides the effect of other gene at different gene loci. So we have already discussed the dominant epistasis and recessive epistasis. We also have different types of epistasis aside from dominant and recessive. It is the double dominant epistasis, the double recessive epistasis, the dominant recessive interaction, and the double gene with cumulative effect. So we will discuss this one by one. But first, let's just have a recap or review on the dominant epistasis. So example here is when the B is epistatic to when the B is the epistatic gene and then the A is the hypostatic gene. So we will have a ratio of 12:3:1 if heterozygous parents are crossed with each other. So in here, you will have a 12 white, 3 yellow, and 1 green. So white is 12 because there is the presence of the B, which is the epistatic gene, the presence of the dominant B. So either it's homozygous or heterozygous, it's not it will produce a masking effect. So it masks the color of this trait. So we'll have a white. So the white colored is 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So if there is a recessive B, so you will have the color effect. So it's colored yellow. So the colored yellow is 1, 2, 3. And then lastly, for the green, it's only 1 because both are recessive. For the recessive epistasis, when C is the epistatic gene and B is the hypostatic gene, you will produce a 9:4:3 ratio when heterozygous parents are crossed with each other. So in here, you will have a black rodents because of the presence or absence of the recessive trait. So in here, you will have the masking effect. The C is the epistatic gene. So with the presence of the recessive C, you will have the masking effect. So we'll have no colored na rodent. So this is an albino rodent. So the ratio is 9:4:3. So another example or type of epistasis is the duplicate dominant epistasis. So this is described as if a dominant allele of both gene loci produces the same phenotype without cumulative effect, and only one dominant gene of either allele is needed to produce the phenotype. So in the absence of all of the dominant genes, the recessive phenotype will be expressed. So the ratio will be 15:1. So in this example, you will have a dominant phenotype because of the presence of the dominant allele, either A or B. So in here, you will produce a dominant phenotype. Also in here, you will also produce a dominant phenotype. But if it's recessive, you will produce a recessive phenotype. So let's have an example. So in here, duplicate dominant epistasis. Because of the presence of both the T and the V dominant, you will produce a triangular shaped seed. So the presence of the absence of the dominant trait, you will produce an ovate seed. So either lang, so T or V. So either of that, when present, you will have a phenotypic expression. Another type is the duplicate recessive epistasis. So this is if both gene loci have homozygous recessive alleles and both of them will produce identical phenotype. So the ratio will be 9:7. So in here, you will have no phenotypic expression in here because there is the presence of the recessive trait. So because of this, you will have no phenotype. But if there is the absence or the absence of the recessive gene, so you will produce a phenotype. Example is this. So this one, you will produce a 9:7 ratio. So the trait, you will have a colorless precursor. So the pigment that will change or catalyze this is coded via this allele. So this allele A will produce a color change which is the same. So colorless pa din ang precursor. And then this one will be converted into purple pigment via this allele B. So A and B allele should be present to produce a purple pigment. So as you can see here, the presence of the A and the B either, you will produce a purple pigment. But the presence of recessive either A or either B, you will produce a colorless precursor because you remove or you cut this chain. So you will produce a colorless pigment. So the ratio will be 9:7. So let's count 1, 2, 3, 4, 5, 6, 7, 8, 9 purple. And then for the white or colorless, because there is the presence of the recessive trait B or recessive A, the frequency is 1, 2, 3, 4, 5, 6, 7. So that's duplicate recessive epistasis. So again, just to reiterate, in this case, the dominant alleles on both locus are required. Hence, wherever A and B both are present, they will result into a purple effect masking the white. This is because the A and B alleles modify the colorless precursor by showing their effect. So, this one, just a hypothetical explanation, the white pigment will be converted to white pigment again via the enzyme Z, which is coded by the C gene. And then this white pigment will be converted into purple gene via the enzyme P, which is coded by the P gene. So, you will produce a 9:7 ratio. 9 purple and 7 white. So, another type of epistasis is the duplicate genes with cumulative effect. So, both the dominant non-allelic alleles, when present together, give a new phenotype, but when allowed to express independently, they give their own phenotypic expression separately. So, in the absence of any dominant allele, the recessive allele is expressed. So, the ratio will be 9:6:1. So, in here, the presence of A or the dominant A, you will only express the A phenotype. The presence of dominant B, either homozygous or heterozygous, you will only produce B phenotype. If it's recessive A and recessive B, so neither A or B is expressed, so recessive trait is expressed. But when there is a presence of dominant A, either homozygous or heterozygous, and the presence of dominant B gene, either homozygous or heterozygous, you will produce a new phenotype. So example is this. So the presence of the A and B dominant, so you will produce a disc shape. So when they are crossed, you will produce a 9:3 or 9:6:1 ratio. So the disc, which is a new phenotype, is A and B. So the presence of the dominant A and B, you will produce a disc shape. So the frequency is 1, 2, 3, 4, 5, 6, 7, 8, 9. But the absence of the B dominant, you will only express the sphere. So in here, the frequency is 1, 2, 3, 4, 5, 6. But if it's recessive to both, you will produce a long shape. So that's duplicate genes with cumulative effect. The last type of epistasis is dominant recessive interaction. So this is the dominant allele either in homozygous or heterozygous condition of one gene, and the homozygous recessive of other gene produce the same phenotype. So the ratio will be 13:3. So example of this is this white leghorn and the white plymouth rock. So when crossed, they will produce this 13:3 ratio. So the colored gene is produced because there is recessive gene. So when there is recessive gene, either if the C is homozygous or heterozygous, you will produce a colored type of trait. So this one is 3. 1, 2, 3. The rest will be 13, which is white. So another topic is pleiotropy. So pleiotropy is the phenomenon in which a single gene conditions affects several or different phenotypical traits. So in this one, the single gene, so we will have pleiotropy or multiple traits. It can produce trait A, trait B, or trait C. Example of this is this chicken, or the fowl. So the dominant frizzle gene produce a feathers that curled outward rather than lying flat against their bodies. So this frizzle gene caused the chicken to have an abnormal body temperature. So these are the multiple traits produced by a single gene. So there can be abnormal body temperature, higher metabolic and blood flow rates, and greater digestive capacity. In contrast to that of the chicken who had feathers that are lying flat against their body. Another example is the sickle cell disease. So in sickle cell anemia, this is an abnormality in the expression of a single mutated HBB gene which produces numerous consequences throughout the body. So the mutated hemoglobin forms polymers and clumps together, causing the deoxygenated sickle red blood cells to assume the disfigured sickle cell. So this one is the disfigured sickle cell. So it's not the typical shape of the red blood cell that is biconcave. So as a result, the cells are inflexible and cannot easily flow through the blood vessels, increasing the risk of blood clots and possibly depriving vital organs of oxygen. So some complications associated with sickle cell anemia include pain, damaged organs, stroke, high blood pressure, and loss of vision. So sickle red blood cells also have a shortened lifespan, and some will die prematurely. Another example is the phenylketonuria. So the phenylketonuria is caused by a defect in the phenylalanine dehydroxylase enzyme. So the gene responsible for that is mutated. So what happens is there can be a downstream problems with the metabolism of phenylalanine. So the patient or individual can have cretinism, albinism, and tyrosinosis. So in here, albinism will be produced because phenylalanine will be converted into tyrosine, and then tyrosine will be converted into melanin. So another is cretinism, and the mental retardation, which causes mental retardation, because phenylalanine will be accumulated or will accumulate in the brain, causing mental retardation. So just to reiterate, cretinism, these are lack of thyroxine from birth or before birth. This could be from lack of thyroid gland or lack of iodine in the mother. It can cause severe and irreparable mental defects, stunted growth, and reduced growth and function of many organs. And then this one is albinism. We also have the lethal genes. So these are genes with at least one allele that, when present in the genotype of an individual, causes death. It can be dominant or recessive lethal. So this one, the perfect example is the Huntington's disease. So if the father is recessive to Huntington's disease and the mother is heterozygous, you will produce a 50% affected child and 50% normal child. So what happens with the brain in the Huntington's disease is this one. So in here, it shows widening of the ventricles as compared to the normal brain of an individual. So you will have a defect. So this causes lethal problems to the patient or individual. So just to summarize, these are the frequency or the ratio for different epistases. So for the dominant epistasis, you will produce a 12:3:1 ratio. For the recessive epistasis, you will have 9:3:4 ratio. For the duplicate recessive genes, 9:7. For the duplicate dominant genes, 15:1. For the duplicate genes with cumulative effect, it's 9:6:1. And lastly, for the dominant recessive interaction epistasis, is 13:3. So that would be all. Thank you for listening and have a great day ahead.